Okay, some of you will be watching this are in Intro to Biology. Others of you watching this will be in Bioinformatics. The emphasis is a little different for the two of you. Those of you in Intro to Biology pretty much need to know, well, everything about this movie, and I'll be going through the things you need to focus on. Those of you in bioinformatics need to focus on the transcription factors, what they're going to call switches here, and others are going to need to focus on basically the history of Darwin, that sort of thing. Now, both of you have learned at least something about transcription factors and the development of organisms from a single-celled organism, what we call differentiation and development. You should try to consider those things as you watch this movie. What they call a switch is a transcription factor binding site in the promoter, and what they call a boss gene is what we would call a transcription factor. So keep that in mind. Transcription factors are proteins, but they're not proteins that make up the stuff of our body, like hair. Hair is a protein, and there's a gene for making it. It's a physical thing. Transcription factors are proteins that pretty much stay inside the nucleus and bind in the promoter region of a gene and control those genes. So they are the controllers of the genes that make up, the rest of the cell, and even are excreted to make things like hair. But in other words, the cells that do work are what we traditionally think of as a gene. But there are genes that make proteins that just stick to the DNA and regulate other genes, and those are called transcription factors, and they bind in that promoter region that we've learned about. Now, those of you, both classes need to learn that part of this video, but some of you need to learn also the history of Darwin. You need to learn about the fish called Tiktaalik, things like that. But both sides also need to learn the four scientists at the end. So let's kind of go through this. Here they're talking early in the movie about where Darwin got his ideas. Basically,
just like many of his modern followers, including evolutionary biologist Sean Carroll.
And this is Sean Carroll. He is not
I developed my interest in animals.
However, he is the pioneering scientist that kind of showed us that transcription factors are really the where all the action is, as they kind of mention in this in this video, is that he's the one who actually showed that, hey, there are genes that control other genes, and he did it in the laboratory. Theory said that they should exist, but no one actually proved it until he demonstrated what we show in this video with the flies. So I would suggest when we talk about Sean Carroll, the people in bioinformatics should also pay attention. You would understand the lab work a little bit better than the people in 1408. 1406, sorry. So please pay attention. You should watch the whole movie, both classes, but there are the 1406 people. There are several places that you should pay very close attention to. They will be on the exam. The people in bioinformatics, you primarily need to understand the parts with the transcription factors and the boss genes, the switches, and the four scientists at the end. The 1406 people will need to pretty much know the whole movie.
It's the same way I think most biologists did, which is either going out in the backyard or going to zoos.
And anytime I got a chance, I'd flip over logs and look for salamanders and snakes and frogs and things like this. And I was just fascinated with their patterns and behavior.
So it was with the young Charles Darwin.
Young Charles liked the traipse around the outdoors. He loved to collect beetles and things. He was a completely ordinary kid.
They're talking to him, so those of you in 1406 will need to watch that part. Now, you will also need to...
companion, somebody to share
coast of Brazil.
The voyage. This is again 1406 people. You will need to know about the voyage, and specifically the fossils in Brazil. You will need to know all about
It was in Argentina that he made his first important discovery.
All right, so make sure you watch that. We're going to jump ahead. Now we get to the Galapagos Islands.
Allowed him to ride on their backs as they slowly lumbered around.
To the giant turtles.
I frequently got on that.
The finches. The finches are very famous. They demonstrate that all of these birds are essentially the same species. They're just slightly different. Of course, depending on your definition of a species, these might be all different species. But ultimately, when you look at their DNA, they all had a common ancestor. Probably they can all breed with one another. So by our definition, they're the same species. They just look a little bit different. They've adapted a bit to each island.
He learns that
now the other thing
whales were descended from creatures with teeth.
A little too far there.
Of nature.
The embryos. When they talk about embryos, this is about development and differentiation. So we talked a little bit about that, and there's a video that's included with the last test in 1406 and just recently in bioinformatics. So there's some talk about embryos. Probably both classes ought to watch the...
Watching a developing embryo is truly the most glorious miracle of nature. I mean, no baloney.
What Darwin learned from studying the embryos amazed him. In snake embryos, you could see tiny bumps, the bony rudiments of legs.
All right, so this is all important to understand. All right, so they begin to talk about here the tree of life and how all organisms are connected.
Ultimately, one type of creature could be transformed into something utterly different. It's a process Darwin called descent with modification. But it all begged a question. Why?
Here is
what was
our tree of life.
Making creatures change?
1406, we need to know.
Darwin needed clues, and he found them in a very surprising place. Dogs.
Dog breeding. Definitely you need to know about dog breeding in 1406 and the how that influenced Darwin.
Big. Small.
All right, we'll jump ahead here a little bit. We're going to talk about the finches some more.
So on a different...
Make sure you watch that. Let's see here. Now here is where they begin to talk about DNA, so this is the stuff
DNA is a code, and its double strand contains all the information to make living things grow and develop.
And that's key, develop.
Lined along each DNA molecule, arranged special sequences of this code that form our genes. Many genes get translated into proteins. And these proteins make the stuff of our bodies. One protein makes hair. Another makes cartilage. Others make muscle.
what makes DNA so amazing is that it just contains 4 letters. But all sorts of combinations of those 4 letters contains all the information for making all the creatures that are on the planet.
It's a gene that determines whether our eyes are blue or not. Another gives us freckles. Another gives us dimples. But DNA has one other vital quality. It doesn't stay the same.
All right. So both classes need to watch this one, especially this part where they're beginning to kind of talk about development. Notice they explained the idea that there are genes that make the stuff of our body, all right, the stuff of the cells, or the stuff that the cells excrete, like that make fingernails and hair. But then there are other types of genes, and they're going to get into that. They're going to talk about what we know to be transcription factors. Now, when this was made, yes, those terms existed, transcription factors and transcription factor binding sites. Those were, and they knew about promoters, but they didn't know until this guy, Carroll, came along. They hadn't proven their theories, and he proved their theories, and we also didn't know how important they were. It was the Human Genome Project, which they will talk about in here, that made us realize that most of the differences between us are not in the protein coding regions, but are instead in the promoters. And that's where, as they say in this movie, all the action is. All right, so I want you to pay attention when they talk about these things. Okay. Let's see.
Nachman was hunting for.
Now here's where they begin looking for tectonic. This is
mutation and evolution. Then an idea took hold. To understand how evolution works, all you need to do is compare creatures' genes.
So here they talked that everyone thought early on that humans would have about 80,000 genes because we were so complex. We had this complex brain and all of that, and we don't have that many genes. And it's not how many genes you have. See, they were assuming before the Human Genome Project that we were complex because we had a lot of genes. When we only had about 24,000, I guess we've gotten it up to about 25, a little over 25,000 now that they've found. And it's not that we have so many genes, it's that we have a more complex regulation of those genes. We use them in different ways. A protein can do many things, and if you make more of it or less of it, and you basically use it in many different ways, then you are actually more complex. And that's why humans are as advanced as we are. It's not because we have more genes, it's because we use them in a more complex way, especially in our brains and our immune systems. Humans have extraordinarily complex brain, of course, neurons. Our neurons and the networks and how the neurons are formed and how they form structures in our brain, extremely complex compared to other organisms, even to chimps. And then, in addition to that, we also have a fairly complex immune system. Now, there are some organisms that have probably more complex immune system than we do, but our immune system is also pretty complex and pretty diverse. It does some strange things. Diet is another one where we're reasonably complex. We can eat a lot of different things.
So when the final answer came in 2003, it was a shocker. 23,000 genes.
Now about 25,000.
The same number as a chicken. Less than an ear of corn.
Plants have many.
People were freaked out by the relatively small number of genes. It's down to something like 22 or 23,000 protein coding genes in the human genome. The simple nematode worm has about that same number. And there are plants that have considerably more genes than the glorious human genome.
The whole Human Genome Project has been a humbling experience as we've discovered that actually it doesn't take as many genes to make a human as we'd all hoped.
And it wasn't just that we had so few genes, but many of our key genes were identical to those of other animals.
And that is the key. We use the same proteins, same genes as all the other animals. We're not that different. We're not that different from a chimp. We're not that different from a fish or really sometimes even a plant. It's not that our proteins are different. I shouldn't say that we're not different. Our genes are not different. But how we use them is different, all right? In other words, you can take the gene for making the fur on a chimp, and it's almost identical to the gene that makes hair on humans. You can take it from a rabbit or a cat or a dog, and it would work just fine in a human, or ours would work fine in theirs. They're not that different. The differences are in the promoters of those genes. The promoters of those genes are very different, so the hair shows up in different places. It shows up in different lengths, different colors, all right? And that's a product of mutations in the promoter region, not in the genes themselves usually. In fact, mutations in the actual protein coding genes, any major mutations usually end up with the organism dying or having some really, really bad disease. And in fact, most of our diseases we think are in the promoter region. Those regions change quickly, and they're usually minor. Things like, minor, but diabetes, things like that. Those are in mutations from the promoter region, usually not in genes. Though you could get diabetes from just not making the insulin, but of course that would be a mutation in the transcription factor, in the RNA polymerase binding site, so that you don't ever make the RNA and make the protein. And so yes, even diabetes is primarily a mutation in the promoter. Now you could make the protein wrong and it wouldn't stick to the outside of the cell in diabetes. That potentially would, of course, lead to diabetes as well. There are many ways to get diabetes, but most of them are mutations in the promoter, not in the actual protein or gene that makes insulin.
What the embryo is telling us, and we have rediscovered what Darwin was talking about all along, that the embryo is where the action is in terms of animal diversity. It is the platform for diversity.
And that is all controlled by the promoter region. In other words, when you use the genes, where and how. All right, so we're going to jump up. Now, here's the part that the bioinformatics class needs to pay attention to, are the parts with flies. You will probably understand it. They begin talking about our boss genes, which are transcription factors, and our transcription factor binding sites, which are just the promoter. They call those regions switches. That's the binding sites, and then boss genes are what they call transcription factors, and they begin to talk about it.
In search of answers, Carol turned to one of the least understood regions of DNA, the vast stretches that were once known as junk. It has been called the dark matter of the genome. Mysterious, uncharted, strange. The vast bulk of the double helix, some 98% of it, doesn't code for proteins.
Now that should stand out as remarkable to everybody, that 98% of the genome doesn't code for proteins. That should have been a warning sign to early biologists, and they knew this. They knew this before we actually sequenced the genome, that majority of it did appear to be non-functional from their perspective. That should have been a warning sign that, hey, something's up here. An organism doesn't waste all that length. And that's what they thought. They just thought that was unused DNA. But the majority of that DNA, as we go further and further into understanding how all of this regulation works, almost all of it is used. There are still some pieces we don't know if they're used or not, but even the places where we think there have been insertions of what we call transposable elements, which are considered to be semi-pathogenic, come to find out they actually are quite useful in evolution, but we don't have time in this course or either course to actually talk about how that works. But probably it's these pathogenic or semi-pathogenic structures called ALU elements, or jumping genes is what they're called sometimes, that are responsible for the evolution of the human brain. But unfortunately, we do not have time to get into that in either class.
Which make the stuff of our bodies. The genes which do comprise just 2%. Even now, no one is sure what much of this huge non-coding area actually does. But it has long beckoned evolutionary detectives like Sean Carroll.
So, this is the band.
So that's the fragment to test.
Yeah.
Carroll had already learned that the paintbrush gene itself was identical in the two types of fly. So he extended his search through their DNA.
Now, both classes need to know what happened to the fly. All right? I'm just going to expect more of the bioinformatics class than I am of the 1406 class.
And in one place,
let's jump up ahead.
Lopsided pattern.
All right.
They concentrated on a group of genes known to control the growth of birds' faces.
Okay, so that would be a transcription factor. A transcription factor would be something that controls development and how you put the other genes together.
As they looked, they saw something intriguing.
In other words, when I say put together, I mean how you use them during development to make yourself different. Why is someone's arms longer than another? Well, the transcription factors are slightly different. The binding sites are slightly different. Maybe, and this will actually go quite a ways to explain that, if that's a little confusing. So make sure you watch this. All right.
Show for their efforts.
Now here they're beginning to talk about Tiktaalik, the fish. Now I'm not going to ask bioinformatics any questions on this, but 1406, you should know this.
Fourth trip seemed pointless. Sequence of the same genes makes our fingers and toes. This was a massive revelation. Suddenly, the origin of creatures with arms and legs didn't seem such a huge leap after all. If the same genes were at work in Tiktaalik, then many of the genes needed to make legs and arms were already being carried around by prehistoric fish. All it needed was a few mutations, a few changes to the timing and order of what was turned off and on, and a fin could become a limb.
All right, now this is going through the idea that you don't have to do a lot in a transcription factor. You don't have to change much in a promoter or perhaps even change a transcription factor itself much to get some pretty major changes in some organisms. The genes are already there. Essentially all life, all vertebrates anyhow, pretty much use the same genes. We pretty much have all the same genes that, you know, things like a chicken has. And therefore, really, the differences between us are just how we use those genes. And that's the important part and the important message here.
Oftentimes, the origin of whole new structures in evolution doesn't involve the origin of new genes or whole new genetic recipes. Old genes, old genetic pathways can be reconfigured to make marvelously wonderful new things.
All right. Moving ahead. Here we
human behavior and the way this orangutan behaved.
And this is where the stuff for bioinformatics.
When Darwin suggested that human beings must actually be descended from apes, he was savaged.
Four particular
he was accused
scientists. One of them.
And one answer
is
all four
thumb
fingers.
Guy, this guy. You need to know what he discovered. All right. So
and that allows us to make grips like this. Gives us a lot of precision.
Is the muscle guy.
As a result of...
So this is the muscle guy. You need to know what he did. All right. So thumb guy, muscle guy. All right. And then there is this guy.
Never thought that I'd be studying evolution. I'm a neurologist interested in the brain and in kids with neurological problems.
So the guy who studies kids' brains, you need to know what he did.
How you doing, buddy? You doing all right?
And then finally, the last one is this lady.
And so we use it.
And she's a bioinformaticist, and she is the one who I will want to point out to both classes. We've talked about what CPG islands are. Notice when they flash these sequences that are important up there, notice how many of them have C's and G's in them. That's very important. Just as an aside, we know what those importances are. Back when this was made, they were just figuring this out. They didn't realize how important CPG islands are, but they are very important. And
technique, which is to look for places where human is different from chimp, but chimp looks almost identical to the other animals.
Alright, so
she too is looking for DNA relating to the human brain.
The brain is one of the things that's changed the most during human evolution, both in terms of its complexity and its size. And so when we look to find the parts of our genome that make us human, we're particularly interested in finding out whether these are things that are involved in the brain.
All right, so you kind of know what she does. All right.
18 letters.
Here are your C's and G's. Notice this. So what do you think she found? If she found a piece that was extra, it's all about turning genes on and off, C's and G's, and hey, that might even be those of you in bioinformatics, that might count as a promoter sequence, the GC box. You never know.
A massive mutation.
All right. And where is it? It's in the promoter.
I think if Darwin were here today, he'd be absolutely stunned, delighted, even moved to see how much his theory has grown.
The interesting thing that they point out at the end is that how well all these new discoveries fit into what Darwin noticed. Now, Darwin was technically not a scientist. To be a scientist, you need to do experiments. And Darwin did no experiments. He was purely observational. This makes Darwin a natural historian. But the amazing thing is that from his simple observation, Darwin, came up with an extraordinary theory to explain everything. Now, he didn't know the details. He didn't know about DNA. He didn't even know about genetics. They had not yet, even though that was coming quickly, it had not been published, and no one, maybe by the end of Darwin's life, it had been discovered, but no one knew about it. No one knew Mendel's laws or anything like that. It's amazing that Darwin put it all together and did so well doing it. And that's what this video is talking about. He put together, he found all the important parts, and he essentially used those parts to say, here's what we know, here's how it may work. I want to emphasize that unless somebody invents a time machine or they do a really long-term experiment over like a million years or something, it would be very hard to prove in the standard statistical way with an experiment that evolution exists. A time machine would do it, but there's lots of evidence for the concept of evolution. One thing we can prove, and this will be a question probably on both of your assignments, both 1406 and in bioinformatics. What we can prove is natural selection. Natural selection is provable in a laboratory. You can basically put a group of bacteria under a stress, say UV light, and basically they will change. You can actually observe over 6 weeks, a year, new genes developing to protect the bacteria from UV light. So keep that in mind, that we can actually see adaptation, which is natural, and the natural selection going on in a laboratory. We have observed it many, many times, and we've observed it many, many times in nature. But the idea here is what happens if you let natural selection go on for millions of years. That's when you get evolution. Unfortunately, we cannot show that natural selection leads to speciation, which is what evolution says. Evolution is simply you take this concept of natural selection and then you let it go on for a long period of time, and from that you get all the species we see on the planet. Evolution is nothing more than that. However, it's controversial because it says that, hey, we're related to monkeys, and that might be contrary to your religion and what we've been taught. It also is kind of says we're not so different from everything else. Humans have a tendency to claim that they're superior and different, and this show kind of points out that we aren't so different. In fact, we're very closely related to a lot of species, and even recently we are very closely related to the apes. It's not that long ago, or the Neanderthals, which were probably human for all intents and purposes. But the idea here is that how different are we? Well, what we developed was a special tool, our large brains, and basically those large brains do make us quite a bit different than any other species on the planet. But that is a very recent development in the long history of life on this planet, and it can be shown that this truly is, through the sequence, the differences between us and all the other animals, including the chimps. They have a larger brain, but they don't have a brain that is complex or as well organized as ours, and that needs to be taken into account. We're still evolving, if you want to think of it that way. The stressors on us are less than they would have been, but certainly humans are still changing. There's a good chance that these ALU elements that I've been talking about are what caused the rapid evolution of our brains. But those have been turned off in our recent history, so our evolution of our brains is kind of stabilized, but stressors can turn those transposable elements, those jumping genes, back on. So when an organism is under stress, the jumping genes tend to be activated. So it actually speeds up evolution, which just throws a huge monkey wrench into everything. And no, Darwin did not anticipate that. He did not anticipate that there could be something like a transposable element which could be turned on and off based on stressors to an organism. So it means you lead a nice, comfortable life. You have plenty of food. Or I should say, just the right amount. Realize too much food can be a stressor as well. So by say obesity or sugar or something like that in modern life might actually activate these ALU elements and increase the mutation rate in the promoters for genes that have to do with the brain. So all of those things would be, it's interesting. Still under development. A lot of this, what I just mentioned here at the end, is still somewhat controversial, but it seems pretty clear that these jumping genes are important nevertheless. Now, what activates them? We're working on that. Something called epigenetics. I wish we had had time to talk about it in both classes. But I do want you to watch this movie. This movie pretty much puts it all together. It connects all these things that we've learned about development and differentiation, CPG islands, and transcription factors. It puts them all together with the development of the human, the sequencing of the human genome and other organisms, puts it all together and kind of gives you a nice package and shows you just how important this is for understanding how, not only how we work, but for medicine alone. This discovery has shown that the majority of diseases are no longer, we don't think that they're mutations in the protein coding genes. The majority of diseases are based in that non-coding region. And so when you do genomic medicine, it's developing. We once thought, well, we're just going to look at the genes, we're going to see how the protein coding regions are different, and we're going to fix them, and that's it. No, it's not so simple. It's much, much more complicated. And that is definitely where medicine is headed. It's just, we're not there yet. All right? But in the future, it should be able to do this. Technologies such as CRISPR will allow us to alter these things, and we may mess it up. I'm sure we will. That's the history of humankind. But we'll learn, and we usually move on and figure it out. The alteration of ourselves, though, is a pretty scary prospect. In other words, in the future, using CRISPR technology, genetic engineering, you will be able to alter things like, even today, it's very reasonable that if somebody wanted to, they could change their eye color with a few treatments from CRISPR. Of course, we've also learned that, yeah, you can change it, and you can actually, in some cases, cure things like cancer with it. You can also cause cancer with it when you don't know how it works. But they say that they're getting much better at the CRISPR, and they're beginning to understand how it causes the cancer when they do inject it into an organism. So yes, they can change your hair color, their eye color. They'll be able to do a lot more if they use CRISPR before birth, when the organism is just a few cells old, or maybe just a single fertilized egg. You could use CRISPR on it, and you could pretty much turn that organism into anything you want. You've seen Planet of the Apes or something. You could definitely do something like that, altering the promoter regions that control the development of the organism. There are so many things, and so many scary things, but also exciting things, curing diseases, things like that we could do. But watch this movie. This is a very good movie. It may take 2 watches. I know it's rather long. It's an hour and 50 minutes. But you should watch it and take notes on the things I've talked about as you watch the movie. All right, and enjoy. It's actually a very watchable movie. It's if you're interested in biology, this is a good movie. All right.